Hi friends, so now I am back with my next video, and in this video we are going to talk about transcription in prokaryotes. So in my previous video I have talked about the replication, the different steps of replication, initiation, elongation and termination, and also the basic process like from the replication is the production of the multiple copies of DNA, and then transcription is the formation of RNA, and translation is the formation of protein from RNA. So please make sure to like, share and subscribe my channel. So what is transcription? So as I have told already, it's a process in which RNA is synthesized using DNA as a template. So here you can see the DNA, which is a double-stranded molecule, and then it get transcribed to form the single-stranded RNA molecule. So I have already talked in my previous video the difference between the DNA, the sugar, which is 2-deoxyribose, and in the RNA, which is a ribose sugar. And here you can see the DNA is double stranded but the RNA is single stranded. So this process is called the transcription. And so what is RNA? So it's a polymer of ribonucleotide which is held by the 3,5 type phosphodiester bridge and are single stranded. It's already in my previous video, so please make sure, if you want to know more, watch the previous video. And it is a molecule which is involved in both functioning and which is involved in both storage, that it can store that information in the form of mRNA, and it can also help in catalysis using the by providing RNAs, the ribosomal RNAs and transfer RNAs for the protein synthesis, the translation process, and it is also involved in the transmission of genetic information. And there are basically three kinds of RNA: the mRNA, which transfers the information in the transmission step, which transfers the information from the gene to the ribosomes, and then transfer RNA, which basically provides amino acids to mRNA and which codes actually the base sequence on mRNA and provide amino acids to form protein. And ribosomal RNA, which is a part of the ribosome. So ribosome is a subunit or the protein subunit which is playing important role in translation. So RNA is a constituent of these ribosomes. So here, as I have already said, different type of RNA and how they are linked, and they are single stranded. So what is the basic difference between the replication, which is the production of making multiple copies of DNA, and the transcription, which is the formation of RNA from DNA? First important difference is the replication. In case of DNA replication, the template is both strands. Like both the strands can act as a template, but in the transcription only transcription is always single stranded. And then you can replicate the whole genome, but in case of transcription only a small portion of genome is transcribed. So you can transcribe multiple portions, but in one go there is only a small portion which will be recognized, and then it will be terminated. For replication you require primers to initiate the replication, but for transcription there is no need for primer. And the enzymes are a bit different. For replication you need DNA polymerase, but for transcription you need RNA polymerase. And the product is, for replication it's double stranded DNA, and for transcription is single stranded RNA. And then the base pairs which are present in both of them. So you have ATGC, in which AT are purines and GC are pyrimidines, right? But like A pairs with T and G pairs with C. But in case of the transcription, instead of T you have uracil. Instead of thymine we have uracil, and then the rest is the same. So what are the features of transcription? So first is it is highly selective. Like transcription cannot happen at any place. It always finds selective nucleotides which are present on the DNA, and only at that position the transcription will be initiated. So this is very selective. And specific sequences, as I have said, it marks the beginning and end of the transcription. Like it decides, the sequence itself describes where the transcription will start and where it will finish. And the signal instructs the enzyme where to start, stop the transcription, when to start. Like if you see, there is a enhancers and repressors which regulates the transcription, so they itself instructs when to start, where to start, and how often to start. So that's why transcription is very it's very regulated process which depends on multiple factors. And the many of the RNA transcripts that are synthesized in the beginning, they are called the primary transcripts, and they can be modified using like different modification, post translation modification, and trimmed, like the 3 ends are converted or different exons to form the functional RNA. So not the primary transcripts is not, it's not always the functional RNA. Functional RNA is formed by the modification of the primary transcript. So where does it occur? In prokaryotes, transcription mainly occurs in the cytoplasm, but in eukaryotes it's occur in the different parts of the nucleus. And the RNA synthesis is mainly catalyzed by different, like one large enzyme which is called the DNA dependent RNA polymerase or RNA polymerase or RNAP. So in case of prokaryotes and eukaryotes, this RNAP, RNA polymerase, in prokaryotes there is only one RNA polymerase which we will talk about in this video, but in eukaryotes there are different kind of RNA polymerase which transcribe different like RNAs like mRNA, rRNA, and tRNA. So this video is particularly focused on prokaryotes, but in my next video I will talk about eukaryotes and different RNA polymerase involved there. So first step, like the strand of the DNA which is transcribed. So you, as you know, in DNA you have the two strands, the 5 to 3 strand and 3 to 5 strand. The strand which is transcribed, like in this case this one, because RNA is always formed in the direction of 5 to 3, right? So that the strand which needs to be transcribed is 3 to 5, because as you know they are anti-parallel. So when the 3 to 5 strand is transcribed, it forms 5 to 3. So 3 to 5 strand is called the template strand, and then another strand is called the coding strand or the sense strand or anti-sense strand. So this is very important to understand what is coding strand, what is template strands, what is sense, anti-sense, because it needs to be converted, like it forms a strand which is exactly as the coding strand but with U. That's why it's called antisense, and what is formed is this. This is what is called RNA, which can form a protein later on. So we will talk about the RNA polymerase. It's an enzyme which play multiple function in the transcription process, like it search and bind to the promoter sites, it unwind a short strand of the helical strand like DNA, so that the proteins, different proteins can bind, and then it can form single stranded RNA. It select the ribonucleotide and the formation of the phosphodiester, as I have talked in my previous video, and it also detect the termination signal. So as you can see, single enzyme playing different role, because it contains different subunits in itself which are performing all this function, and it also interact with the activator and repressor that regulates the transcription. So there are all 3 stages of transcription in prokaryotes. First is the initiation phase, where the RNA polymerase recognizes the promoter and starts the transcription. The elongation phase, the RNA is continuously growing, and then the termination phase, where the polymerization stops and the RNA that is formed is separated from the DNA template. So I will talk about all these 3. So first step is the initiation. So how does the enzyme know where to bind, right? So you know, it's a long strand of ribonucleotides or deoxyribonucleotides in case of DNA. So enzyme is lost where to bind, but then the strand itself provide the information that there are basically different factors which are present, or different sequence which are present. So this is one of the sequence TGTGGACA, which is present as -35, and then there is one of this box which is called Tata box because of the sequence, which are present as -10. So it means that if the transcription is starting here, if the start point is here, 10 bases away from it is minus 10 and 35 is minus 35. So there are some sequences of 6 to 8 nucleotides which are present, which are signals for the enzyme to bind and starts the transcription. So if the sequence is present in prokaryotes, then the enzyme will bind and then it will start the transcription. This is only for prokaryotes. For eukaryotes, it's a bit different. So once the enzyme finds this sequence, the RNA polymerase, it binds here and with the sigma factor and starts to open this strand and forms the open complex. So now it's the same like straight line, which is called the close complex because they are intertwined with each other. But then the polymerase starts unwinding these two strands, and it forms what is called an open complex. So here you can see now that we have the start site and that where the RNA polymerase bind. So once it opens this complex, then starts the elongation phase. So now once this complex is open, different nucleotides starts to add it, ribonucleotide starts to add, and sigma factor is released. And then the enzyme moves in the forward direction and continue producing more RNA until it finds a stop signal. So once it finds a stop signal, I will explain how it get terminated and release of the RNA. So what is termination? It basically depends on the two termination signal, the row dependent termination and row independent termination. So we will talk about the row dependent termination. So once the RNA is formed, so this is the place where the cannot bind. So this is the place where it's starting, right? And then it find a RUT sequence, RUT site, which is a continuation of different nucleotides, GC and AT content, which are so many. And when it finds these, it gets the enzyme, the factor row, which binds to this site, and then it moves forward. It depends on the ATP for its function, right? So they are inverted repeats and they are recognized by the termination protein, which is called the row factor. And once it works in a ATP dependent manner, right, it binds to the signal and then it starts to move to the RNAP, and then RNAP, the RNA polymerase, cannot move further, so it dissociates from the RNA and release the nascent RNA as well. So this is the way where RNA, like the row dependent termination work, because it recognizes this site and then row factor comes. But there are some cases in which this is row independent. So it is like the palindromics repeats of GC. So palindromic means like here you can see it reads the same from this side and this side. So once the strand once there are these palindromic repeats, they are like self complementary, right, as you can see here, and they can form a hairpin structure, and this is the signal to stop the transcription and termination. And then the second feature is a huge repeats of A residues that are transcribed into U, and then the RNA transcription just stops there when you have this multiple of U residues. So I hope it's clear the transcription in prokaryotes, how it initiates, elongate and terminate. And if you have any doubts, please let me know in the comment section, and please make sure to like, share and subscribe my channel. Thank you.